Okay, so let's get into the fun part of the kidneys, which is reabsorption and filtration in the renal tubule. So, first, the structure. So, if you read the slides, you'll have this idea of what the structure of the renal tubule looks like. We know we start at the renal corpuscle, which we just went over in the previous video. And now we're in the renal tubule. So we're making the filtrate in the corpuscle, and then we're passing the filtrate through this tubule. And I've drawn it all spread out, but it's really all curled up on itself. So this part here is all twisted and turned, and this part here is all twisted and turned, and this part is actually way up close to this efferent arteriole. It's like right next to it, so it's completely like looped around on itself. But one thing that is kind of an accurate representation is the way that this little dip, this loop here, goes down deeper into the medulla or the center of the kidney. So we've got the filtrate passing in this direction always, going from the corpuscle out. So the sections of the renal tubule are first the PCT, or the proximal convoluted tubule. Then it's going to flow down into this loop here, which is the loop of Henle. The loop of Henle goes down on this side, that's the descending limb, and then it goes up on this side, that's the ascending limb. And both of these limbs are going to have what are called thick segments and thin segment at the bottom here. So a little more on that in a second. First, let's go up out of the ascending limb of the loop of Henle. We'll go into the distal convoluted tubule, or the DCT, then finally into a collecting duct. Now once we're out of the collecting duct, so once the filtrate passes out of the collecting duct, there's going to be no more filtration or reabsorption, but we're just going to pass it through a series of tubules until it finally goes out of your body as urine. So from a collecting duct, a bunch of collecting ducts are going to dump into a papillary duct, which goes out through the renal pyramids, right down the center of the papilla, which is why it's called a papillary duct, which is the papilla is the point of the renal pyramid. From there, coming out of that papillary duct, it goes into a minor calyx. When you have a few of these minor calyces flowing together, they form a major calyx, and a couple of major calyces coming together form the renal pelvis. And the renal pelvis goes out through the hilus of the kidney, which is the entrance to the kidney, the door, if you think of it that way, like the opening where things like the renal pelvis are going out, the renal vein is going out, the renal artery is going in through the hilus. But the renal pelvis is carrying the filtrate, which is now essentially urine, out of the kidney into a ureter, which leads to the bladder, which is where it will be stored until micturition, which is urination, and that is occurring through the urethra. So that is the path that our filtrate takes. So just back to the structure of our renal tubule. You'll notice that I have put these little square blocky things around most of the renal tubule, around everything except for the thin segment of the loop of Henle. These are the peritubular cells, and those would be wrapped around the renal tubule. They completely surround the renal tubule. And in the case where I put them as these little sort of blocky cells, they are simple cuboidal epithelial cells. Now, where I haven't put the cells, we still have peritubular cells, but they're going to be simple squamous epithelial cells, so they're much, much flatter. This is going to have a big effect on permeability. So where we have the simple cuboidal epithelium, we're going to have a lot of ions able to move across between the cells and the filtrate, and then between the cells and the peritubular capillaries, which are the capillaries that are spread all over the renal tubule. So ions and water will be able to move across the cells between the filtrate and the blood, where we have simple cuboidal epithelium. And we'll get into some detail as to which ions are going to be able to move where and how. Now, when we have this situation down here where we have the thin loop of Henle with just simple squamous epithelium, then we're not going to have ions able to pass here. Only water can pass between what's in the filtrate and what's in the blood here. So no ion movement here at all. And then when we get back up here and we have our simple cuboidal again, we can resume ion and water movement here. Okay, so this is going to become important. So make sure that you really think about that. As we have simple cuboidal epithelium, ions and water are able to interact, move from the filtrate into the cells or from the cells into the filtrate. Or on the other side of the cells, we can have ions and water moving from the cells into the blood or from the blood into the cells, always crossing a cell membrane. This cell membrane and this cell membrane. So ions and water moving wherever we have simple cuboidal epithelium, which is everywhere, except for the thin segment. Then in the thin segment, only water can pass between the cells and the blood. So only water is able to move out of the filtrate here. Now, one more structural thing, in addition to the paratubular capillaries, which are just kind of splatted all over the entire renal tubule, we also have a capillary that flows downwards alongside the loop of Henle, and this is called the vasa recta. So this is going in towards the medulla. So from the cortex, which up here would be the cortex, so everything from here up would be the cortex, or the outer part of the kidney. The loop itself is in the medulla. So the vasa recta is going down towards the medulla. And that's going to be important in our next video, which goes over reabsorption and secretion.